So in lecture, we finished up talking with the last slide of titration and how we can use it to quantify how many antibodies that a patient has. Again, really looking to see how strong is their immune system if exposed to something such as chickenpox in a lab. Another of these immune system type tests is called a neutralization test. And what we're looking for is, does a patient have an antibody that could potentially neutralize a virus or a toxin? And how we know if it neutralizes a virus or a toxin is because we know viruses and toxins will kill cells. It's called the cytopathic effect. It just means that they're going to kill cells. However, if a patient would have an antibody to neutralize that virus or toxin, then it can no longer kill the cell. So it's called the viral neutralization test. So how it works, I've got a nice image right here showing this. So we know if we expose a toxin to a cell, the toxin's going to kill the cell. That's the point of it. However, if a patient has antibodies against that particular toxin, and a patient's serum, their blood serum, is mixed with the toxin, and then in a lab exposed to that same cell, because the toxins are neutralized by that patient's antibody, the cells are going to live. Same idea as if we're looking to see if this virus will infect these red blood cells. If we add the patient's antibodies, and the patient has the antibodies in their blood serum, the patient's antibodies are going to attach to the virus, neutralizing it so that the red blood cells don't get infected, they don't get destroyed. What that can look like in a lab underneath a microscope so we can see or visually see the cells is we can study and look at the cells excuse me, underneath the microscope over time. We can see in this particular effect, this is going from several hours. I think it's read every 6 hours. Lots of very healthy cells, but then over that next 24, even up to 48 hours, we can see cells dying. That would indicate the patient did not have the antibodies and that the virus or the toxin that was added to these cells ultimately killed the cells. So that can say, nope, patient doesn't have antibodies against that virus or toxin. Or if all the cells looked like this 24 to 48 hours later, say, yes, the patient does have the antibodies. It's another kind of fun, tricky way to, to identify if a patient has antibodies or not. But again, it's not the only way. Again, there's multiple ways we can detect antibodies or detect the presence of antigens. Another way is called a labeled antibody test. So we can take an antibody, and the antibodies always look like Ys, and we can actually tag them in a lab. So on the little body part, again, I think of these two as the hands, and then the rest is the body. We can actually put a little tag on the body because the body's not specific for things shaped wise. So we can tag it, and what we can tag it with is we can tag it with dyes, we can tag it with enzymes, we can tag it with something that's going to glow like a radioactive chemical. So we can tag and add different things to these little bodies of the antibodies. So when they talk about labeled antibody tests, they're talking about labeling or tagging that body portion of the antibody. And we can use these little tags to then either detect antigens or antibodies. Because if an antibody has a tag on it that, say, will glow under UV light, if the antibody is present because it's stuck to what it's supposed to stick to, we can now see that easily glowing underneath a UV light. And we have microscopes that we have UV lights that we add right to it. One very common antibody test that's done that uses these labeled antibodies is called an ELISA, which stands for enzyme-linked immunosorbent assay. Is that the little label that we tag onto that antibody is an enzyme. And my recall from a couple weeks ago, when you mix an enzyme and a substrate that's specific for that enzyme, when you mix them together, the two together will actually produce a color change. So we're looking to see if the enzyme is present and we add a substrate, color will happen. So by looking and detecting that presence of the enzyme, we're really looking and detecting the presence of that enzyme-linked antibody, that it's attaching to what it's supposed to. And it can actually even help us not just detect an antibody or an antigen, we can even quantify it, because the brighter or darker the color, the more of the enzyme was able to stick to something. And it's extremely sensitive, meaning 1 or 2 antigens and it will stick, and you're going to see somewhat of a color change. Even just looking at this, all of these are individual samples from like 96 different patients we can detect, and we can see that everyone that shades a blue is considered positive. It means the enzyme was present because it stuck to what it was supposed to, what we're looking for. But we can also see some of the blues are darker blue and some of them are lighter blues, so it helps us somewhat quantify it as well. Now, how the enzyme or the ELISA test works. There is a direct test and an indirect test. So we're going to first, a direct test again is just a refresher, is we are looking for the presence of an antigen. So we're looking for an antigen. So I'm going to show that base over in this little well. What we do is this little plastic well, this is showing this little indent, this little well on this plate. They coat it with, I should say that first they're going to add antibodies that are specific for this particular antigen. So we're detecting for this antigen. We'll call it chickenpox, the chickenpox virus. So here's the chickenpox virus. So we're trying to determine if the patient in their blood serum has the chickenpox virus. So we're going to add an antibody that we know attaches and is specific for the chickenpox virus. We're going to add that antibody to the inside of these plate wells. And these plate wells are kind of sticky. And then we add a gelatin so that nothing else sticks that we don't want to, except these antibodies that are specific for chickenpox. Then we're going to add the patient's serum. And if the patient has the chickenpox virus, right here, it's going to stick to this antibody. If the patient doesn't have the chickenpox virus, well, nothing's going to stick here because they don't have anything that's specific for this antibody. Then we're going to add that enzyme linked. So here's that little tag or label on the body of this antibody. And so here's an antibody that is specific for chickenpox, and we've tagged it, we've labeled it with an enzyme. And because this patient had chickenpox, so it's stuck to this antibody, and because it's here stuck, this antibody is going to see it attached to it as well. So we've made a little sandwich here of antibodies. An antibody that we know binds to chickenpox, and an antibody that we've enzyme linked. A little enzyme on there that also recognizes chickenpox. Then we're going to add a colorless substrate. However, if this colorless substrate comes into contact with this enzyme that's present, that's when we get these color changes. So the only way that this color change is going to happen is because this patient has that antigen, they have that virus, the chickenpox, or whatever else we're testing for. If the patient did not have this virus, nothing would be stuck to this antibody, which means this thing's not going to stick to it, and it actually gets rinsed out in the process, and it would show up as colorless. That's how we can detect the antigen, so direct test. Well, we can also do this as an indirect test as well. Let's just say we're going to test for antibodies against chickenpox. So first we're going to actually put chickenpox antigen right on the plate well. So we'll just say here's our virus right here. We attach it to the plate well. The plate wells, these are a sticky kind of substance. And then we coat it with something so that nothing else sticks to it. Then we're going to add the patient's serum. And we're going to assume that this, not assume, but we're going to make it so this patient does have antibodies against chickenpox. So their little red antibodies that are specific for chickenpox are going to attach to that chickenpox that's stuck on the plate well. But then we're going to add an enzyme-linked antibodies. And we actually have enzyme-linked antibodies that can recognize other antibodies. So they don't recognize any particular specific antibody. They just see the body portion of an antibody, because that's not really specific. They just see the body portion of an antibody and they attach to it. So we have enzyme-linked an antibody that can recognize antibodies. And yes, it's called an anti-antibody. So because the patient has the antibody, our enzyme-linked antibody recognizes it and attaches. We add that colorless substrate. Because this enzyme's present, we get a color, indicating that that patient had the antibody. So ELISAs can be done quickly, cheaply, because we can truly detect 96 different patients, all for chickenpox or whatever it is we're testing for, Or we could take one person, and in each of these wells, we detect a completely different thing. We could test one patient to get 96 different things. And we have machines that even do this for us now that, add all the reagents and suck them up and rinse everything out, and then even analyze the colors that appear as well. It's great. Now, the Western blot test is similar to the ELISA, meaning we're still adding antibodies and antigens, and because of that enzyme that's attached to the antibodies, we're looking for color changes. But the biggest difference is that we use a Western blot when we're looking not just for one specific antigen. We're looking for multiple antigens, and we want to sort out the antigens by size. So we can detect antibodies against multiple different antigens. Again, we're just going to separate them out by size. So we are going to take a gel. This looks very similar to the agar we use in lab, except it's clear and not red. And in that agar, in that gel, we have little indents or divots that we can add patient samples to. We subject it to electricity. The electricity causes the separation of antigens. The smaller the antigen, the farther and easier they can kind of wiggle through that gel and make it. So the biggest antigens are stuck. They don't travel very far. The tiniest ones separate and move quite far when subjected to electricity. So we just separate the antigens out by size in a patient sample. Then, it's called the blot for a reason, is we need to get these antigens we separated by size from the gel onto paper. And so this is where the blotting comes in, is they take, it says absorbent paper, truly just a sponge, kitchen sponge. We take that sponge, we get it super, super wet. We put the gel that we just got done that has all those separated antigens on it. We put the paper that we want it to go on, and then we take filter paper, or usually just a stack of paper towel, and we squish it down. And the idea is if you take a stack of paper towel and you squish it down on a wet sponge, the water is going to go from the sponge onto the paper towel. However, in the process of going from sponge up into paper towel, as the water travels, it's going to pick up the antigens from the gel, deposit them on that paper as they go up into the paper towel. So we're just using the movement of water. So now we have our paper that has all of our antigens on it, all separated by size. We add the enzyme-linked antibody to it, and we get our color, nice bright color changes on our paper. Now, why they do this, and the most common time that they actually use the Western blot, is they test for Lyme's disease. So the CDC requires 5 out of 10 antigen bands to say that someone actually has Lyme's disease. So it's not just saying, oh, well, they have one band, they must have Lyme's disease. CDC is like, well, no, because that particular antigen size-wise, maybe it was a broken fragment, but it's not enough to say that that particular bacteria is present in the body. It says, well, we need to see at least 5 out of the 10 confirmed antigens that Lyme's disease have, and we separate them out by size. So my column 1 here, this is a negative control to make sure we didn't have any contamination. My column 8 is the positive control, and we're looking for any patients. We have a patient 2, 3, 4, 5, 6, and 7. We're looking to see which of their patients, 2 through 7, actually have Lyme's disease. As long as they have 5 out of the 10 bands showing, we conclude them as being positive for Lyme's disease. So in our area, which is prevalent for Lyme's disease, this is the most common time Western blots are done. So we can see patient 3 absolutely has Lyme's disease, patient 2 as well. If we go back and look at these patients, here's our negative and positive control. Definitely patient 3 has Lyme's disease, patient 5 as well, and I would even say patient 6. These lines are faint, but they are still present. Then our last types of tests are point of care testing. These are the super quick immune system tests that gives results in minutes, and it should look very similar to what we've done in lab with that rapid strep test. So some of them use, it's called the immunochromatography assay. Rapid strep, that's, I was going to say pregnancy tests, rapid strep tests. It's the exact same test that's done when they test for mono. That looks all these different things. That way, add that little test strip in, and as everything moves up from the patient sample, all of their antigens are going to meet with antibodies, and you look for the particular color change that can occur. Immunofiltration is we're not looking to see that movement of antigens going up the strip. Instead, they're just looking for a color change that's happening right on the spot. So sometimes it's even called a monospot test, just because of that quick little spot they do on there. One of the common tests that they use this immunofiltration assay for with the filter is testing for chlamydia. Gets really quick, easy results. So it's just same kind of basic ideas. It's that antigen-antibody meeting. We just get really fast, distinct color reactions to give us those quick results. So that's the end of this PowerPoint lecture. So make sure you take the quiz on Chapter 17.